good morning to all of you. Am I audible to all of you? Please write in the box. Yes, good morning. Good morning, everyone. Am I audible to all of you? Once again, a very good morning to all of you. Do you hear me? Okay, let's start with the topic, that is detoxification. Do you hear me? The box. Am I audible to all of you? Yes. Today we will discuss a very important topic, that is metabolism of xenobiotics. It is called as biotransformation, detoxification, detoxic. It is a very important point of view. Okay, am I audible to all of you? Please write in the box. Priyanka, Apeksha, Mansi, am I audible to all of you? Okay. So let's start. What is xenobiotics? Xenobiotics, this is a Greek word composed by two words, xenos plus biotics. Xenos means stranger. What does it mean? Xenos means stranger, and biotic, you all know, life. So a xenobiotic is a compound that is foreign to the body. A xenobiotic is a foreign chemical substance found within an organism that is not normally naturally produced by or expected to be present within. So it means that is not normally naturally present or produced or expected to be present within the body. So metabolism of xenobiotics is also known as metabolism of foreign compounds. Metabolism of xenobiotics is also called as metabolism of foreign compounds. Please add in the box. Is it clear to all of you? Xenos means stranger or the compound that is foreign to the body. So a xenobiotic is a foreign chemical substance which is an organism which is found in an organism that is not normally naturally produced by or expected to be present within. Okay. Increasingly, humans are subjected to exposure to various foreign chemicals or compounds, xenobiotics like drugs, food additives, pollutants, you all know. Drugs, food additives, and pollutants. Do you agree with this statement? Drugs, food additives, and pollutants. These substances are increasingly exposure. Next, what is detoxification and other is detoxification of biochemical reactions occurring to convert foreign, which is often toxic compound, to non-toxic or less toxic, more easily excretable form, which are mainly eliminated through urine and bile. So the series or the various series of biochemical reactions occurring in the body which convert foreign compounds to non-toxic or less toxically excretable form. It is called as detoxification or detoxification. This word detoxification is misleading sometimes. Why it is so? Because detoxified products may be more toxic than original form, but are more stable, more soluble, polar, and easily excretable compounds. These can be more toxic but more water soluble and easily. Examples are methanol converted into formaldehyde, enogens can be converted into carcinogens, which are more toxic than originally. Is it so? Then aromatic ones. Examples are oxidation of methyl group containing compounds. Methyl group is oxidized to acid through formation of alcohol and aldehyde. So oxidation of alcohols, primary aliphatic and aromatic alcohols are oxidized to corresponding acids. Oxidation of alcohols, primary aliphatic and aromatic alcohols. Like methanol, you all know what is the formula or chemical formula of methanol. Please write in the box. What is the structure of methanol or methyl alcohol? Please write in the box. Yes. CH3OH, formaldehyde. Please write in the box the formula of formaldehyde, HCHO. And what is formic acid? Please write down the structure of formic acid. Yes, HCOOH. Likewise, ethanol is converted into acetaldehyde and then it converts into acetic acid. CH3, CH2OH or C2H5OH, CH3CHO, and this is CH3COOH. So is it clear to all of you? Benzyl alcohol is converted into benzaldehyde and then this benzaldehyde converts into benzoic acid. This is methanol, formic acid HCOH, ethanol C2H5OH converts into acetic acid CH3COH, benzyl alcohol C6H5CH2OH converts into benzoic acid C6H5COOH. So aldehyde are oxidized to produce corresponding acids like benzaldehyde. You all know benzaldehyde converts into OOH. Amines and their derivatives. Aliphatic amines are converted to the corresponding acids, liberating urea, while aromatic amino acids are oxidized to phenol. You can see here aliphatic amines, then it converts into aliphatic acids and urea. This aniline converts into para-amino phenol. This is para-amino phenol. Then aromatic hydrocarbons, benzenes may be oxidized to mono, di, or trihydroxyphenols as shown below. Benzene is converted into phenol OH, 2 quinol, catechol, and hydroxyquinol 1, 2, and 3. Sulfur compounds. Organic sulfur is oxidized to sulfuric acid. Drugs. Meprobamate is a tranquilizer. It is oxidized to hydroxy meprobamate and excreted in urine. So you can see here oxidation of drugs, meprobamate, this hydroxy meprobamate, and this chloral is converted into trichloroacetic acid. What is methanol toxicity? You have heard this word methanol toxicity, and this is also called as detoxification. You know this methanol by the enzyme alcohol dehydrogenase converted into formaldehyde, and this converts into HCOH, that is formic acid. Methanol, formaldehyde, formic acid, and then CO2 and H2. So methanol has a relatively low toxicity. Methanol is metabolized in the liver. In the first step of degradation, methanol is transformed to formaldehyde via the enzyme alcohol dehydrogenase. The main biocatalyst the main enzyme involved in this reaction is alcohol dehydrogenase. This is also known as ADH. So transformation of formaldehyde to formic acid via the enzyme aldehyde dehydrogenase is faster. So the metabolism of formic acid is very slow. It often accumulates in the body, which results in the metabolic acidosis. The major damage occurs to the optic nerve, you all know. So ethanol is the antidote of this methanol toxicity. Since it is the true substrate of alcohol dehydrogenase, methanol is spared. So which is the antidote of methanol toxicity? Please write in the box. Which is the antidote of methanol toxicity? Yes, ethanol. This methanol converts into formaldehyde and then it converts into formic acid. Second reaction is reduction. A few examples of detoxification by reduction are given, like picric acid converts into picramic acid, chloral hydrate converts into trichloroethanol, nitrobenzene converts into aminobenzene. This C6H5NO2 converts into aminobenzene C6H5NH2. This trichloral hydrate converts into trichloroethanol OH2 CCl3 CH2OH. This picric acid, you all know, 3 NO2s, and then it converts into picramic acid C6H2OH NO2 NH2. Next reaction is hydrolysis. The hydrolysis of the bonds such as ester, glycoside, and amide is important in the metabolism of xenobiotics. Several compounds undergo hydrolysis during the course of their detoxification. These include aspirin, acetanilide, diisopropyl fluorophosphate, atropine, and procaine. These are the important reactions. What is the meaning of hydrolysis? Please write in the box. What does it mean? Hydrolysis. Please write in the box. What does it mean? What is the meaning of this word hydrolysis? Please write in the box. Am I audible? Am I audible to all of you? Please write in the box. What is the meaning of the word hydrolysis? Sakshi, Avinash. Yes. Hydrolysis means addition of. What does it mean? Hydrolysis. Yes, addition of water. So these are the examples of detoxification by hydrolysis. First, as I told you, aspirin, acetyl salicylic acid converts into salicylic acid and acetic acid. Acetanilide, diisopropyl fluorophosphate DFP. It is a nerve gas. You all know. Atropine converts into tropic acid, tropine, procaine. It can be used for anesthesia. Converts into para-aminobenzoic acid and diethylaminoethanol. Digitalis converts into sugar and A-glycan. And this detoxification of arsenic, mercury, cadmium. These are the foreign compounds to the body. It is called as xenobiotics. The compound BAL. What is BAL? British anti-levisite, which was used in Second World War, is important compound for the detoxification of these metals, arsenic, mercury, and cadmium. BAL plus metal excreted in urine. These metals are excreted easily with BAL. BAL. What is the full form of this BAL? A distance for British anti-leviside, which was used in Second World War. Active enzyme with cysteine at active site plus heavy metal converts into inactive enzyme plus BAL. This is the antidote for these heavy metals, arsenic, mercury, and cadmium, which was used in Second World War. So BAL stands for, please write in the box, BAL stands for BAL. BAL, B-A-L. What does it mean? And BAL stands for, please write in the box, BAL, B-A-L. Am I audible to all of you? Okay, British anti-leviside. So this is aspirin, acetyl salicylic acid, you all know, converts into by the addition of H2O, salicylic acid and acetic acid. Likewise, acetaldehyde with H2O, aniline plus acetic acid. Then diisopropyl fluorophosphate with addition of H2O converts into dialkyl phosphate. This atropine converts into tropic acid and tropane, and this procane is converted into para-aminobenzoic acid and diethylamino ethanol with the addition of water or H2O. This is the reaction. Procane with water converts into para-aminobenzoic acid. So these are the enzymes which are involved in phase 1 reactions like hydrolysis esterase peptidase, which are present in microsomes, cytosomes, lysosomes. Reduction involves azonitro reduction, carbonyl reduction, disulfide reduction. Likewise, oxidation, which is most important, most common, most frequent reaction of phase 1 is oxidation. It includes alcohol dehydrogenase, aldehyde dehydrogenase, aldehyde oxidase, xanthine oxidase. You all know, you have studied many of these enzymes, and the most important is cytochrome P450. The most important enzyme is cytochrome P450, which is a microsomal enzyme and also present in mitochondria. So the most important enzyme of oxidation is cytochrome P450, mainly catalyzed by members of class of enzyme referred to as monooxygenases. So there are three names: monooxygenases, mixed function oxidases, or cytochrome P450. Mixed function oxidases, cytochrome P450, monooxygenases. Other enzymes of significance are aldehyde and alcohol dehydrogenase, deaminases, you all know, esterases, amidases, and epoxide hydroxylases. So the important category of enzymes of oxidation, enzymes involved in oxidation are monooxygenase, mixed function oxidase, or cytochrome P450. So cytochrome P450 enzyme system, the reaction catalyzed by this monooxygenase cytochrome P450 is as follows. This is RH, you all know, by the addition of oxygen you can get ROH plus H2O plus NADP. RH can represent a wide range of xenobiotics, including drugs, carcinogens, pesticides, petroleum products, and pollutants. In addition to these exogenous or foreign substances, endogenous compounds such as certain steroids, eicosanoids, fatty acids, retinoids, and also various substances. These substances are generally lipophilic and are rendered more hydrophilic by hydroxylation. By the process of hydroxylation, we have just studied. Most of the reaction of cytochrome P450 involves the addition of hydroxyl group to aliphatic or aromatic compounds, which may be represented as above reaction like this. RH plus oxygen, NADPH plus hydrogen, H plus, and then gives or it converts into ROH plus H2O plus NADP. So what is the role of cytochrome 450? Please write in the box the names of cytochrome P450. Please write in the box the names of cytochrome P450. Am I audible to all of you? Yes, first is monooxygenases. Second one is mixed function oxidases. Monooxygenases, mixed function oxidases, or cytochrome P450. So what are the role, or what is the role of cytochrome P450? The uses P450, this P450 refers to the absorption peak, that is 4 at 450 nm. So it is called as cytochrome P450, exhibited by the enzyme when exposed to carbon monoxide. So cytochrome P450 refers to the absorption peak at 450 nm. They are present in highest amount in liver and small intestine, but are probably present in all tissues. In liver and other tissues, they are present mainly in the membranes of smooth endoplasmic reticulum. Smooth endoplasmic reticulum, we can say in microsomes. These are small portion of small endoplasmic reticulum or microsomes. In the adrenal, they are found in mitochondria as well as in ER. Approximately 50% of the drug are metabolized by the isoforms of cytochrome P450, or we can say mixed functional oxidases. So please remember these names: monooxygenases, mixed functional oxidases, and cytochrome P450. Approximately 50% of the drugs are metabolized by the isoforms of cytochrome P450. So it is important for the metabolism of drugs, or we can say metabolism of xenobiotics. Is it? Please write in the box. Is it clear? What are the silent features of cytochrome P450? This is an important question. This is the important question. Silent features or characteristic features of cytochrome P450. Multiple forms of cytochrome P450 are believed to be exist, ranging from 20 to 200. At least 6 isoforms of cytochrome P450 have been isolated yet. They are all hemoproteins. These are all hemoproteins. Cytochrome A, cytochrome AA3, do you remember cytochrome A? Cytochrome AA3, where we have read this cytochrome A? Yes, where? Yes, in ETC, electron transport chain or respiratory chains. Okay, so these are hemoproteins containing heme as a prosthetic group. Cytochrome P450 species are found in the highest concentration in microsomes of liver, as I earlier told you. In the adrenal gland, they occur in mitochondria. The mechanism of action of cytochrome P450 is complex and dependent on NADPH. Where we have read this word NADPH, please write in the box. In which cycle we have studied this NADPH or NADP? Please write in the box. NADP. Yes, we have read in HMP pathway. Exose monophosphate shunt. We have read this word NADP. Okay, very good. The phospholipid phosphatidylcholine is a constituent of cytochrome P450 system, which is necessary for the action of this enzyme. So phospholipid phosphatidylcholine. Phosphatidylcholine is a constituent of this cytochrome P450. Cytochrome P450 is an inducible enzyme. Why it is called as inducible enzymes? because its synthesis is increased by the administration of drugs such as phenobarbital. So it is called as inducible enzyme because its synthesis is increased by the administration of drugs such as phenobarbital. So it is called as inducible enzyme. A distinct species, namely cytochrome P448, 448 with absorption peak at 448 nm has been studied. It is specific for the metabolism of polycyclic aromatic hydrocarbon, hence it is also known as aromatic hydrocarbon hydroxylase. So which one is aromatic hydrocarbon hydroxylase? Please write in the box. Which one is aromatic hydrocarbon hydroxylase? Please write in the box. P448. Some exhibit genetic polymorphism which can result in mode of action of cytochrome P450 via, I just told you, NADPH. This is oxidized NADPH cytochrome P450 reductase. This is reduced NADPH cytochrome P450 reductase. So this converts into NADP plus. Here you can see this reduced cytochrome P450 is converted into, this oxidized cytochrome P450 is converted into reduced cytochrome P450 by this enzyme, and this is hydroxylated Xenobiotics XOH, and this is oxidized NADPH cytochrome P450 reductase, and this is reduced cytochrome P450, and this is oxidized this is hydroxylated Xenobiotics, this is XOH. This XH is converted into XOH. This is the mode of action of cytochrome P450. It is also called as monooxygenases and mixed function oxidases. Phase 2. So, in phase 1, what are the reactions of phase 1? Phase 1 includes oxidation, reduction, and please write in the box. Phase 1 includes, please write in the box, which reactions? Please write in the box. Oxidation, reduction, and hydrolysis. Overall hydroxylation of the xenobiotic compounds. So phase 2, which includes conjugation. This is the most important reaction after this oxidation. Conjugation is the important reaction of this whole process, metabolism of xenobiotics, or we can say biotransformation or detoxification or detoxification. Conjugation is a process by which the foreign molecules and their metabolites are coupled with a conjugating agent and are converted to soluble non-toxic derivatives which are easily excreted in urine. So, conjugation is a process by which the foreign molecule and their metabolites are coupled. These are coupled with a conjugated agent, and by this conjugation, it is converted into soluble non-toxic derivatives which are easily excreted in urine. Conjugation reaction can occur independently or can follow phase 1 hydroxylation reaction. You all know oxidation. The most frequent and most common reaction is oxidation. After this oxidation, the conjugation reaction occurs. Conjugation takes place primarily in liver but can occur in kidney also. After conjugation, the products are generally rendered non-toxic, but in certain conditions, they are left unchanged or become more toxic. We have just read, and toxification at least 8 different conjugation agents have been identified in the body. These are glucuronic acid, glycine, cysteine of glutathione, glutamine, methyl group, sulfate, acetic acid, and thiosulfates. These are the conjugating agents. Type of phase 2 reactions means conjugation includes glucuronidation, sulfation, acetylation, methylation, conjugation with amino acid, and conjugation with glutathione, that is GSH. So glucuronidation, sulfation, acetylation, methylation, conjugation with amino acids, conjugation with glutathione. These are the important reactions which include in conjugation phase 2 reactions of metabolism of xenobiotics. First is glucuronidation. You all know glucuronidation is the most frequent conjugation reaction. UDP glucuronic acid is the glucuronyl donor which is formed in the uronic acid pathway of glucose metabolism. So UDP glucuronic acid is the glucuronyl donor which is formed in the uronic acid pathway of glucose metabolism. Microsomal enzyme glucuronosyl transferase present in both endoplasmic reticulum, that is microsomal, and cytosol are the catalyst for this reaction. The glucuronide may be attached to oxygen, nitrogen, or sulfur groups of the substrates. Compounds conjugated with glucuronic acids are bilirubin, you all know, aromatic acids, benzoic acid, phenol, secondary and tertiary aliphatic alcohols like phenyl glucuronic forms. In this reaction, phenyl glucuronide forms. Then antibiotics like chloramphenicol, hormones, thyroid hormone, derivatives of corticosteroids, and sex hormone metabolites. 2-acetyl aminofluorine, a carcinogen, aniline, then meprobamate, a tranquilizer. Certain drugs like barbiturates, when administered, induce glucuronide transferase, and this increases the glucuronide formation. So, glucuronic acid conjugation may occur with compound containing hydroxyl, carbonyl, sulfhydryl, or amino groups. In general, conjugation with glucuronic acid produces strongly acidic compounds which are more water soluble or hydrophilic at physiological pH, therefore are easily excreted in urine. So these are the compounds conjugated with glucuronic acids: bilirubin, aromatic acids, phenol, antibiotics, hormones, aniline, and certain drugs. Glucuronidation. This UDP glucose, in the presence of this UDP G dehydrogenase, converts into UDP glucuronic acid. This UDP glucose, in the presence of this UDP glucose dehydrogenase, converts into UDP glucuronic acid. And then this UDP glucuronic acid plus bilirubin, means conjugation with bilirubin conjugate with this UDP glucuronic acid, in the presence of this UDP glucuronyl transferase, converts into bilirubin monoglucuronide plus UDP. So this reaction is important. Please remember this reaction. Here you can see this glucuronidation of bilirubin. Bilirubin monoglucuronide plus UDP gluconic acid. Again, the enzyme is UDP glucuronyl transferase. It converts into bilirubin diglucuronide plus UDP. Most of the bilirubin excreted in the bile of mammals is in the form of bilirubin diglucuronide. You all know. This bilirubin UGT activity can be induced by a number of clinically useful drugs like phenobarbital. Next conjugation reaction is sulfation. The sulfate donor is adenosine 3-phosphate 5-phosphosulfate, that is PAPS, PAPS. This is adenosine 3-phosphate 5-phosphosulfate. This compound is called active sulfate. Enzyme is involved in this reaction is sulfotransferase. And compounds which are conjugated with this sulfate are as follows, like phenol, cresol, indol, steroids, estrogen, androgen, tyrosine to form tyrosine O sulfate, which is required for the formation of fibrinogen. Other compounds, glycosaminoglycans, glycolipids, and glycoproteins. This is the reaction. Phenol converts into this active sulfate in the presence of this active sulfate, phenyl sulfuric acid. Next reaction is acetylation. Acetylation is presented by this reaction. You can see here acetyl coenzyme A, and this combines with this X, acetyl X, and coenzyme A, where X represents a xenobiotics or any foreign compound. Acetyl coenzyme A, active acetate, is the acetyl donor. These reactions are catalyzed by the enzyme acetyl transferases present in the cytosol of various tissues, particularly in liver. Compounds conjugated by acetylation like sulfanilamide, PABA, you all know, para-amino benzoic acid, and isonazide. This PABA, para-amino benzoic acid, the presence of this enzyme converts into acetylated PABA, is active acetyl coenzyme A. Acetyl coenzyme A is the active form of acetic acid that takes part in the conjugated reactions. Drugs such as sulfonamides are converted to acetyl derivatives. Sulfonamide plus acetyl coenzyme converts into acetyl sulfanilamide plus coenzyme A. Next reaction is methylation. Methylation is limited in the body. S-adenosyl methionine. Active methionine is S-adenosyl methionine, acts as methyl group donor. Reactions are called as transmethylation reactions, and the enzyme involved in this reaction are methyl transferases. These are the examples of methylation. Compounds conjugated by methylations are nicotinamides, nicotinamide methyl group transfers, and methyl nicotinamides. So likewise, this paramethyl amino azobenzene, addition of methyl group, it converts into para-dimethyl amino azobenzene hepatic carcinogen. Likewise, O-methylation of estrogen, norepinephrine, epinephrine, and their metabolites. Conjugation with amino acids. Likewise, first number, conjugation with glycine. Glycine amino acid, conjugation with glycine amino acid. This benzoic acid plus glycine, you all know. Hippuric acid is formed. Nicotinamide plus glycine, nicotinuric acid. Cholic and deoxycholic acid are conjugated to form glycolic acid and glycodeoxycholic acid. So, benzoic acid plus glycine, where we have studied, please write in the box, benzoic acid plus glycine. Yes, in LFTs. What is this glycolic acid? Please write in the box, glycolic acid. Please write in the box. What are the bile pigments? Name any two bile pigments. Bilirubin and biliverdin. And bile salts are, please write in the box, bile salts. Please write in the box, bile salts. Name the bile salts. Yes. Glycocolate, third. Yes. Okay. Chlorocolate, yes. So next reaction is conjugation with cysteine amino acid. A few aromatic compounds are conjugated with cysteine in the presence of acetic acid to form mercapturic acid. This is an important reaction. So bromobenzene plus cysteine plus acetic acid converts into bromophenyl mercapturic acid, and this naphthalene, cysteine, and acetic acid combines, and this will form naphthyl mercapturic acid. Next is conjugation with glutamine. Phenyl acetic acid plus glutamine converts into phenyl acetyl glutamine. This reaction is important in patients of phenylketonuria. Since excess of phenylacetyl glutamine is excreted in urine, that imparts a mousy odor to the urine. This is an important compound, phenylacetyl glutamine. Because this compound is excreted in the urine of the patients of phenylketonuria. Conjugation with glutathione. What is glutathione? Please write in the box. What is glutathione? Or what is the composition of glutathione? What is glutathione? This is a tripeptide. You all know. This is a tripeptide. Gamma glutamyl cystinyl glycine. Gamma glutamyl cystinyl glycine. This is a tripeptide consisting of glutamic acid, cysteine, and glycine amino acid. It is composed by 3 amino acids. These are glutamic acid, cysteine, and glycine. Glutathione is commonly abbreviated as GSH because of the self-hydryl group of its cysteine, which is the part of this molecule. A number of potentially toxic electrophilic xenobiotics, such as certain carcinogens, are conjugated to the nucleophilic GS in the reaction, GSH in the reaction, that can be represented as follows: R plus GSH, R, S, G. This R is here an electrophilic xenobiotics. So it is important in the case of cancer because a number of potentially toxic electrophilic xenobiotics are conjugated to this nucleophilic GSH in reaction, that can be represented like this: R plus GSH converts into RSG. This nucleophilic GSH, and toxic is electrophilic xenobiotic. The enzyme catalyzing these reactions are called as glutathione S transferases. A variety of glutathione S transferases are present in human tissue. They exhibit different substrate specificities and can be separated by electrophoretic techniques. If the potential toxic xenobiotics were not conjugated to GSH, they would be free to combine covalently with DNA, RNA, or cell protein and could thus lead to serious cell damage or cell toxicity. So GSS is therefore an important defense mechanism against certain toxic compounds such as some drugs and some carcinogens or cancer-producing agents. So this GSH is therefore an important defense mechanism against certain toxic compounds and some drugs and carcinogen compounds. The whole reaction is represented here. A wide range of organic compounds such as alkyl, aryl halides, alkenes, nitro compounds, and epoxides get conjugated with cysteine or glutathione. The formation of mercapturic acid and the glutamate and glycines of glutathione are removed, and acetyl group is added to this cysteine residue. You can see here. This GSS transferase, cysteine glycine, and this R cysteine, and lastly mercapturic acid is formed. This GSS transferase. Gamma glutamyl transpeptidase, this cysteine glycinase, and here an acetyl transferase. Ultimately, it forms mercapturic acid. Conjugated with thiosulfate, the highly toxic cyanoids are conjugated with thiosulfate to form less toxic thiocyanate. So this cyanide, sodium thiosulfate, is less toxic thiocyanate. Cyanide is converted into thiocyanate by the addition of this thiosulfate, sodium thiosulfate. So this cyanide converts into less toxic thiocyanate by the addition of this thiosulfate. Is it clear to all of you? Please add in the box. Is it clear to all of you? Overall, conjugated reactions and their reagents. You can see here, these are the reactions, these are the reagent. Like Glucuronidation reagent is UDP, glucuronate, and the groups are these. Sulfation, PAPS, you all know. In methylation, SAM, acetylation, acetyl coenzyme A, sulfide formation, glutathione, amide formation, glycine and taurine. What is SAM? Please add in the box. What is SAM? In methylation reaction, what is SAM? Please add in the form. SAM stands for. Please add in the box. SAM stands for. S. Adenosyl methionine. This is the active methionine. What is methionine? Please add in the box. S. Adenosyl methionine. Okay, good. So, glucuronidation reagent is UDP glucuronate, sulfation PAPS. What is the full form of PAPS? Please add in the box. PAPS. PAPS. What is the full form of PAPS? Please add in the box. What is the full form of PAPS? You all know. We have just studied. What is PAPS? This is adenosine 3-phosphate 5-phosphosulfate. It is called as PAPS. 3-phosphate 5-phosphosulfate. Sulfate donor adenosine 3-phosphate 5-phosphosulfate. This is active sulfate or a sulfate donor. The overall detoxification or biotransformation pathway. These are the toxins. These are the water-soluble waste products. These are lipophilic in nature and these are hydrophilic. Is it clear to all of you? This phase 1 reaction includes oxidation, reduction, and which one? Please add in the box. Yes, hydrolysis. And phase 2, conjugation. So, these hydroxylation reactions, phase 1, and these conjugation reactions, toxins, metabolic end products, microorganisms, contaminants, pollutants, insecticide, pesticides, food adulterants, drugs, and alcohol. These are xenobiotics. These are xenobiotic toxins, or you can say foreign compounds. These are water-soluble, excretable form, waste products, hydroxyxenobiotics, you can say, elimination from the body via gallbladder, bile, and stool, you all know, and via kidney in the form of in the urine. So it is clear to all of you, this whole process of detoxification or biotransformation. These are the toxins, lipophilic in nature. These are water soluble or hydrophilic waste products, and these are eliminated from the body via gallbladder, kidney, and many other organs. So what is the role of liver in detoxification? Liver detoxifies harmful substances such as alcohol, drugs, toxins, you all know, that have entered the bloodstream by breaking them into harmless forms for excretion. Another is drugs such as antibiotics, penicillin, erythromycin, you all know, and sulfonides are also broken down into harmless ways which are then excreted by the body. Another is hormones such as thyroid hormones, steroid hormones such as estrogen, testosterone, aldosterone are similarly inactivated, ready for removal from the blood through the kidney. So, liver detoxify alcohol, drugs, toxins, antibiotics, and hormones. So is it clear to all of you? Role of liver in detoxification. Liver detoxified hormone substances like alcohol, drugs, toxins, antibiotics like penicillin, erythromycins, and hormones like thyroid hormones, estrogen, steroid hormones, testosterone, aldosterones. Phase 1 and phase 2, liver detoxification. You can again see here, this is fat soluble toxins, oxidation, reduction, hydrolysis, hydration, the various reactions are there. Overall, we can say hydroxylation by cytochrome P450 enzymes. Another name, mixed function oxidases, and these are nutrient needed, vitamin B2, B3, B6, B12 like coenzymes, folic acids, glutathione, flavonoids, etc. And these are by phase 1. Then it enters into phase 2. The compound enters into phase 2 in the conjugation pathway, sulfation, glucuronidation, glutathione conjugation, acetylation, amino acid conjugation, and methylation. These are needed for these reactions like methionine, cysteine, magnesium, glutathione, vitamin B12, vitamin C, glycine, taurine, glutamine, folic acid, and choline. And by these water-soluble waste eliminated via urine, bile, and stool. So is it clear to all of you? This is the overall reaction of phase 1 and phase 2 by liver, or the process of detoxification by liver. Effects of xenobiotics. Xenobiotics can produce a variety of biological effects, including pharmacological responses, toxicity like cell injury, you can say cell toxicity, immunological responses, you can say by producing antibodies, cancer, that is the process is called as carcinogenesis. Some chemicals like benzo alpha pyrene requires activation by monooxygenases in the endoplasmic reticulum to become carcinogenic. So they are thus called as indirect carcinogens. The products of the action of certain monooxygenases or some pro-carcinogens substrate are called as epoxide. It is an important word, epoxide. Epoxides are highly reactive and mutagenic or carcinogenic in nature, or maybe both. Epoxide hydroxylase like cytochrome P450 acts on these compounds, converting them into much less reactive dihydrodiols. So this is the important word, epoxides, the process of cancer. You can see here. Overall effects of xenobiotics are, you can see here, these xenobiotics, these are foreign compounds by the process of cytochrome detoxification or in the presence of cytochrome P450, these reactive metabolites, non-toxic, then again by the process of GSH or glutathione S transferase or epoxide hydroxylase, it converts into non-toxic metabolites. These reactive metabolites converts binding to covalent binding to macromolecules, and then they creates, or they can injure the cells, then produce antibody production by a heptane, and then ultimately cell injury, and then they can, and the mutation can also occur, and this leads to cancer. So ultimately these reactions leads to cell injury, cell toxicity, cell injury, and cell injury, and via mutation leads to cancer. So these are the ultimate effect of xenobiotics. Now I summarize the whole presentation of this metabolism of xenobiotics or detoxification, or we can say biotransformation. Do you understand? Now, is it clear to all of you? Please, box. Am I audible to all of you? Please add in the box. Am I audible? So, please add in the box the reactions or the phases of biotransformation. Quickly, I will summarize the whole presentation. Yes, these are the phases of biotransformation. Phase 1 and phase 2. Which reactions are involved in phase 1? Which reactions are involved in phase 1? Oxidation, next, which is the most common or most frequent pathway of biotransformation? Oxidation followed by conjugation. The right answer is oxidation followed by conjugation. So now I repeat my whole presentation. What is xenobiotics? Xenobiotic is a foreign compound to the body. So it is not normally naturally produced by or expected to present within the body. Metabolism of xenobiotics is metabolic of foreign compounds. What is detoxification or detoxification? This is the series of biochemical reaction occurring in the body to convert often toxic compounds to non-toxic or less toxic, or importantly, more easily excretable form. Then sometimes it can be more toxic than original, like methanol converts into formaldehydes and pro-carcinogens converts into carcinogens. Site of detoxification mainly liver and then lung, kidney, intestine, placenta, brain, skin, and adrenal glands. What is biotransformation? Biotransformation is the process whereby a substance is changed from one chemical form to another chemical form, or we can say it is the conversion of lipophilic to hydrophilic chemicals catalyzed by enzymes in the liver and other tissues. Most important purpose of this biotransformation is lessens the toxicity of xenobiotics. But in certain circumstances, these reactions can still increase the toxicity of a foreign compound. So these are called as intoxification reactions, like methyl intoxification or methanol intoxifications. This is the example of methanol. What is the significance of these biotransformation reactions? Importantly, facilitates excretion, detoxification, and metabolic activation also. Biomedical importance, by pharmacology and toxicology, you can understand it, pharmacy and therapeutics, management of cancer, drug addiction. Compounds that are detoxified includes foreign compounds, compound produced in body which are to be eliminated like bilirubin, steroids, and compound produced in intestine by bacterial putrefaction or fermentation like indole, skatol, etc. These are categorized into two: exogenous, endogenous. Exogenous are drugs, food additives, pollutants, insecticides, carcinogens, and endogenous, bilirubin, bile acids, and steroids. These are the factors affecting biotransformation of drugs: diet, hormonal status, genetics, disease, age, development status, functional status of the liver and kidney. Phase 1 reaction, oxidation, reduction, and hydrolysis. Commonly, they are called as hydroxylation reactions, OH, functional group. And phase 2 is conjugation reaction of phase 1 compounds, or direct conjugation can also occur. So most important thing, oxidation followed by conjugation is most important or most common, most frequent biotransformational reaction in the metabolism of xenobiotics. Overview of biotransformation reaction, this phase 1, phase 2, maybe metabolic activation. This is the difference between phase 1 and phase 2. Important is increase in hydrophilicity by phase 1 small, it is large. Conjugation may result in metabolic activation, it always facilitates excretion. Phase 1 oxidation, a large number of foreign substances are destroyed by oxidation in the body. Methanol converts into formaldehyde, then formic acid, ethanol, acetaldehyde, acetic acid, benzoyl alcohol, benzaldehyde, benzoic acid. Amines and their derivatives. Aliphatic amines converts into aliphatic acids and urea. Aniline converts into para-amino phenol. Likewise, benzene converts into phenol, quinol. Some drugs are oxidized. Meprobamate converts into hydroxy meprobamate. What is methanol toxicity? You all know, and detoxification. And ethanol is the antidote for this methanol toxicity. Enzyme involved in this reaction is alcohol dehydrogenase. This is the whole process. Second is reduction reaction. Picric acid converts into pitramide acid, then chloral trichloroethanol, and this chloral hydrate. This nitrobenzene converts into aminobenzene, addition of hydrogen. Hydrolysis. The hydrolysis of the bonds such as ester, glycosides, and amide is important in metabolism of xenobiotics. These include aspirin, acetaldehyde, diisopropyl fluorophosphate, atropine, and procaine. This is important. BAL, BAL, British anti-levisite, was used in World War II, and this BAL plus metal excited in urine. So the antidote of this heavy metal arsenic, mercury, and cadmium is BAL, British anti-levisite. This diisopropyl fluorophosphate DFP is nerve gas. Aspirin converts into salicylic acid plus acetic acid, and likewise. The chemical structure of this aspirin, acetyl salicylic acid. The addition of H2O or water converts into salicylic acid and acetic acid. Here it again acetaldehyde, H2O, addition of H2O converts into aniline plus acetic acid. Likewise, atropine converts into tropic acid and propane with the addition of H2O, and this procaine with the addition of H2O converts into para-aminobenzoic acid and diethyl aminoethanol. These are some enzymes involved in the process of hydrolysis, reduction, and oxidation, phase 1 reactions, and these are the localization of these enzymes. In hydrolysis, esterase peptidases, reduction, these enzymes, and oxidation, alcohol dehydrogenase, and cytochrome P450 is the most important microsomal enzymes. Phase 1 reaction enzymes are monooxygenases, mixed functional oxidases, or we can say cytochrome P450. This is important because most of the reaction of cytochrome P450 involve the addition of hydroxyl group to aliphatic or aromatic compound, which may be represented like this: RH plus oxygen. This converts into ROH. What is the role of cytochrome P450? Why it is called as cytochrome P450? Please write in the box. Why it is called as P450, cytochrome P450? Why it is known as P450? Because absorption peak is 450 nm. They are present in highest amount in liver and small intestine. In liver, this is mainly present in the membrane or vesicles of smooth endoplasmic reticulum, that is known as microsomes. Approximately 50% of the drug are metabolized by isoform of cytochrome P450. These are the silent features of cytochrome P450. These are also called as inducible enzymes because its synthesis is increased by the administration of drugs such as phenobarbital. This is the mode of action of cytochrome P450 via NADPH or NADP. This is reduced cytochrome P450. This is oxidized cytochrome 450. It converts in the presence of NADPH cytochrome P450 reductase, that is in reduced form. This is Xenobiotics, or this is hydroxylated Xenobiotics. Conjugation, phase 2 reactions. Conjugation is the process by which the foreign molecules and their metabolites are coupled with the conjugated agents and converts to soluble non-toxic derivatives which are easily excreted in urine. These are the reaction: glucuronidation, sulfation, acetylation, methylation, conjugation with various amino acids, and conjugation with glutathione. This glucuronidation, you all know about this glucuronidation, this UDP gluconic acid, and this donor is UDP glucuronic acid. Enzyme is glucuronosyl transferases. This converts into UDP gluconic acid, then with the conjugation with bilirubin, form bilirubin monoglucuronide. And this bilirubin diglucuronide. Most of the bilirubin excreted in the bile of mammals is in the form of bilirubin diglucuronide. Sulfation, you all know active sulfur or sulfate donor is. Active sulfate is PAPS. Phenyl sulfuric acid. Acetylation, the addition of this active acetate. Acetyl donor is acetyl coenzyme A in the presence of acetyl transferases. This acetylate PABA, PAB, acetyl coenzyme A. And another example is sulfenylide plus acetyl coenzyme A, acetyl sulfanilamide plus coenzyme A. Methylation. Methylation is limited in the body, and this by SEM, S-adenosyl methionine, this active methionine acts as methyl group donor. Reactions are called as trimethylation, and the enzyme is methyl transferases. You can see here addition of methyl group. Conjugation with amino acid, first is with glycine. You all know benzoic acid plus glycine combines, forms hippuric acid. And this by cysteine amino acid, you can say here bromophenyl mercapturic acid and naphthyl mercapturic acid. With glutamine, you all know phenyl acetyl glutamine. This is an important product because it is excreted in urine, phenyl acetyl glutamine. Conjugation with glutathione. What is glutathione? This is a tripeptide gamma glutamyl cystenyl glycine consisting of glutamic acid, cysteine, and glycine. Glutathione is commonly abbreviated GSH. Enzyme is glutathionease transferases. It is involved in defense mechanism against certain toxic compounds such as drugs and carcinogens. This is the whole reaction. It converts into mercapturic acid. Conjugated with thiosulfate, this cyanide, toxic Xenobiotics, converts into lesser toxic thiocyanate. These are the conjugated reactions and their reagents. Glucuronidation, UDP gluconate, sulfation PAPS, methylation SEM, acetylation acetyl coenzyme A, sulfide formation glutathione, amide formation glycine and taurine. This is the whole mechanism of the detoxification. These are lipophilic toxins. These are waste products which are water soluble or hydrophilic in nature by phase 1 and phase 2, eliminated from the body via gallbladder, bile and stool, kidneys, urine. Role of liver in detoxification. Liver detoxified harmful substances like alcohol, drugs, toxins, antibiotics, and hormones. This is the important slide. This includes both phase 1 and phase 2 liver detoxification. These are the reactions: oxidation, reduction, hydrolysis, hydration, dehalogenation. Enzymes are cytochrome P450 enzyme. These are lipophilic, fat soluble. These are water soluble, hydrophilic. Phase 2 is the compounds. By phase 1 enters into phase 2. Processes are sulfation, glucuronidation, glutathione conjugation, acetylation, and many more. And by this, more acetated form, water soluble, eliminated via urine, bile, and stool. These are the effect of xenobiotics: pharmacological responses, toxicity, cytotoxicity or cytotoxicity, immunological responses via antibodies and haptens, and lastly, cancer. So ultimately it leads to cell injury, and via mutation leads to cancer or carcinogenesis. Now, is it clear to all of you? Now, is it clear to all of you? Please add in the box. Am I audible to all of you? Okay, so thank you very much.